All right. Assalamu alaikum wa rahmatullah. Peace be upon you all, and welcome to another Finding Truth podcast. And today I have Otangelo Grasso with me, who is a Christian creationist interested in science and especially with the topic of evolution. We have actually had our one debate before about Christianity and Islam, or the nature of Jesus, I think it was. But today we are joining forces to, he will give us a presentation about whether the evidence shows that DNA and RNA are by design or not. So obviously he's arguing it is by design, right, Otangelo? So if you'd like even to give more introduction to yourself, and then we can go to your presentation, I guess.
Yeah, sure. For who doesn't know me, I am an evangelical Christian. I am Swiss Italian, but live in Brazil. And I do apologetics on my Facebook account and also on YouTube, wherever I have the opportunity to join and defend creationism and intelligent design, and also generally about the Christian faith. I am happy to do that.
Great. So let's see what you have for us today in terms of your presentation about the DNA and biogenesis of DNA.
Yes, of course, the topic is not about evolution because DNA is universal in all life forms. So it is a prerequisite to have life starting and having self-replication, and in the end, eventually, also evolution. So the topic is about how the DNA molecule, the biosynthesis pathways, if they are better explained by natural mechanisms or by eventually an intelligent agent which had to be there with his power and intelligence to create actually DNA, RNA, and in the end, all life forms. So this is what this stream is about. So
All right.
what we have here is a DNA molecule, and it is basically made of three parts. That is the nucleobase, the ribose, and the phosphate backbone. They are joined here by bonds, and the origin of the three parts is what we need an explanation for. That means that the origin of the individual parts, the base, the ribose backbone, the sugar, and the phosphate. Okay, so here we have the three parts: the phosphate group, the sugars, and the bases. So in RNA, we have the purine ring, adenine and guanine, and the pyrimidine ring is cytosine and uracil. And DNA has the uracil replaced by thymine. The difference between the DNA and the RNA sugar is this hydrogen, which is removed on DNA. We have here a hydroxyl group and on RNA, and that is removed, and that gives the stability to DNA. So DNA is a much more stable molecule than RNA. RNA disintegrates much faster, and that is what makes DNA a better information storage molecule compared to DNA.
Okay.
And there is another difference here. I am not able to actually, in this format, to read what I have here down. Can you show that?
Down
I
where?
will. Yeah, well, I will just read it here from
Okay.
my other screen here. Okay, so the uracil base in the RNA is replaced with thymine used in DNA, and uracil and thymine differ in just a methyl group. A methyl group has one carbon and three hydrogen atoms attached to it. Now, why is it replaced in DNA? There is a logical reason for that. So the deamination of cytosine is a spontaneous reaction that occurs in aqueous solutions. It means the removal of an amino group at the 4' position, and it is replaced with an oxygen atom. And in the process, ammonia is released. That leads to the formation of uracil. But if uracil is present in DNA, then it would be difficult to know that it is the original uracil or it has been formed by deamination of cytosine. In this way, the mutations occurring in the deamination of cytosine could not be detected, which will lead to undetected errors and undesired mutations and eventually an error catastrophe.
Mhm.
So do you have a kind of doubt or question to that?
Yeah. So what you're indicating here is that the letters in the RNA case are put together in a way so that, because it's an unstable molecule, so that the deamination will not confuse the system, right?
Yes. In DNA we have cytosine, and in RNA we have uracil. So in order for cytosine not deaminate and become uracil, and then the system confusing it, I will explain that in the next shares.
Yeah.
Here we have it. So this creates a problem for the information stored in the genome. If it is in a coding section, then rather than coding for isoleucine, as shown in the picture here, for example, it would code for threonine, which is a problem because it would mutate the amino acid sequence of a protein and eventually compromise its function. So the cell employs sophisticated molecular machinery to avoid this and transforming uracil to thymine. So
Okay.
So the question, of course, would be, why would a non-intelligent, unguided events prebiotically have that foresight and knowledge that this error catastrophe could occur, and then eventually come up with that switching of cytosine, uh, uh, of switching the molecule and having thymine rather than uracil in DNA. So you need foresight in order to prevent this. And if you don't have an intelligence doing that, well, what do you replace it with? Because natural events, they don't have foresight. And you need actually foresight in order to recognize this problem. So this is very strong evidence for intelligent design.
Yeah. So what you're saying here is that the system is protecting itself from an RNA codon not easily traceable to its origin in DNA, right?
Yeah, the system wouldn't recognize that it was, if it is their deamination of cytosine, and then it would basically it would confound the original information with the mutated information, and it would translate the wrong codon. Okay. And of course, that will result in a mutated amino acid, a wrong amino acid insertion in the protein, and eventually that can have drastic consequences. We know of certain amino acids where just one replacement of one amino acid will have a problem in its function. And a very prominent example, it's not an amino acid, but a ribosome RNA, is in the transferase center of the ribosome, where you have the core, where the polymerization reaction occurs. And amongst 2,900 RNAs, there is one specific one which has to be precisely at the correct place. And if it's not there, then you don't have functional polymerization. So there are some proteins where this is very important, that you have the exact right sequence. There are others where it's not so important, but in some it is important. So that's why cells spend such a huge amount of energy to error detect and correct errors. That starts with DNA, and also the ribosome has over 13 different error check and repair mechanisms. So it spends a lot of efforts to error check and correct wrong things. So this is also something which had to emerge prebiotically, and you had to have this minimization of errors right when life started. And that is a very powerful argument for intelligent design, because otherwise, why would natural or non-intelligent mechanism instantiate so complex error check and repair mechanisms?
Yeah.
So here we have another interesting thing, that cells, they do not only make RNA and DNA from scratch, but when they are worn out, then they are degraded and recycled. This is also something very interesting. Why are there recycle mechanisms installed, and how did that emerge prebiotically by non-intelligent, non-guided events? This is a big question mark for those which advocate for abiogenesis. How was this decided, or what was the mechanism that came up with these ingenious solutions which we use? We recycle, use stuff, or degrade it, or throw it away. And we have the same in cells, where we have degradation mechanisms, catabolism, recycle mechanisms. We have the proteasome, which actually cuts amino acids apart and cuts proteins, and then recycles the monomers, the individual amino acids, and also DNA, recycles it. So this is very ingenious solutions which are implemented here, and this is powerful evidence of intelligent design.
Yeah, and we can also think that even between different living things, they are all using, the same material, and this makes it useful for some creatures to feed on other creatures. So at the end of the day, it's all genetic material and proteins. And when you ingest, for example, proteins from another creature, you can use them in your body, and you can also complete the cycle even from the protein level, not only inside the cell from the DNA level, but even between organisms on the protein level and on the carbohydrates level, etc.
This is a good observation, Ahmed, and I think it is really interesting that we are made of basically just 4 basic building blocks, which are the nucleotides, amino acids, carbohydrates, and phospholipids. And based on just these 4 building components, all the different cells and organisms are made. And if you think and consider that, it is really amazing.
Yeah, absolutely. Because sometimes some people would say, well, don't you see that this is an evidence of evolution, that it is the same, DNA and the same RNA elements, and at the end of the day, the same amino acids that are generated from the RNA codons. And then the thing is, it is actually a stronger evidence for a single designer, because otherwise, if we don't have the same 20 amino acids, then if we eat another, all carnivores, if they eat another creature, it might be poisonous because it has amino acids that they have no use for. They cannot recycle them and build their bodies from the proteins of other creatures. And it's also amazing that even plants are using the same DNA coding system, and hence the same proteins. So if it's a herbivore, it eats plants and it builds its body. If it's a carnivore, it eats another animal and builds its body. Bacteria can clean up the whole thing. Phages can do their work on bacteria and kill the bad ones. And so the whole system, from viruses to bacteria to fungi to animals to plants, it's the same genetic base, it's the same RNA, it's the same amino acids, and then everything can integrate and metabolize each other and clean and keep the planet clean at the end of the day. It's the perfect system for recycling.
Yes.
Yeah. So it goes even the whole cycle. Yeah.
Yes, and you have also the energy cycles, like the nitrogen cycle, the oxygen cycle, the water cycle, the sulfur cycle, the rare mineral cycle, and so forth. And if you don't have these materials cycling right from the beginning, you can't have life. But the most interesting thing is that these cycles, they are also requiring bacteria, which drive that cycle. Like the nitrogen cycle is based on bacteria, which are complementary. They complement each other. And if you don't have that cycle, then you cannot fix nitrogen. If you don't have fixed nitrogen, which makes ammonia and nitrate, you cannot incorporate nitrogen into the cells to make amino acids and nucleotides. And then where do you get it from? And these are all the abiogenesis problems. If you don't have that source, where do you get these materials from? And the same goes with carbon, which is the basic building block of life. You need to get it somewhere. And the question is, where did the material come from prebiotically? How do you have organic material prebiotically? And we know that these are also very complex processes where you need to fix carbon dioxide to have carbon. And if you don't have the cycles like the reverse TCA cycle, which is composed of 9 very complex enzymes which produce and fix carbon, where do you get it from? And I mean, those which have these metabolic first scenario proposals, they are left with non-enzymatic mechanisms, and they think that some kind of clay would have generated the first fixed carbon, but they have pointed out that this is too unspecific. You need to have energy. Where do you get funneled energy from to have that these reactions going on? So you run in big problems here. That's why abiogenesis is a big unresolved problem, and most people, they don't have not the slightest clue about the size of that problem. And I think that you have great names and great guests on your channel, like James Tour, I think you had here.
Fazrana also,
and Hugh Gross and Fazali Rana, and I'm certain that they have on a great length and extent elucidated these problems.
Yes, absolutely, and actually, I have gone into very into length with Dr. Fazali Rana, for example, concerning the composition of DNA and RNA, and even at one instance, he made a great explanation of why we even have those specific 20 amino acids, and because there is like 500 of them in nature, but it occurs that it's almost the same 20 across everybody. And it turns out that even those 20 amino acids are specifically an optimum set. So if you make them more than 20 or less than 20, or you try to replace these 20 with another 20, there are some properties of those amino acids that relate to their electrical properties and other properties that will not be an optimum set. So you cannot build all this great diversity coupled with stability. And the same goes with even the letters, the ATCG, and even when you replace it with the U. Also, those 4, we have something concerning parity checks and complementarity of those nucleotides. That's also amazing. So I would really recommend that if you have not been, I'm talking to the audience, if you have not gone through the series, it's called Seeing God in Biology with Dr. Fazrana. I would really recommend that people will go there and go into length into those topics too.
Yeah, and I am actually writing a book on abiogenesis, and what you touched in regards of that is, I was investigating why are they saying that there are 500 amino acids, and actually, what has been discovered is about 300 different amino acids. But then, if you ask yourself, well, why 300? And the thing is that you have basically an unlimited possibility of different side chains. So I would say that in reality, the number of possible amino acids is not just restricted to 300 or 500, but it is in reality unlimited. It's unlimited. And something which I don't think maybe Fazali has not touched to that, but these amino acids, they can come as alpha amino acids or beta amino acids or gamma amino acids, and the ribosome accepts only alpha amino acids. So if you feed it with beta amino acids, they are now doing engineering with ribosomes, and they are able to change the reaction pocket, and then you can feed them with beta amino acids. But the ribosome used in life actually only can be fed with alpha amino acids. So you have there a complementarity where you have the ribosome designed to receive only alpha amino acids, and we have only alpha amino acids in life, while you can have all these different alternative configurations. So this is again fantastic evidence for design, where you have this complementarity of the machine and the feedstock with which the machine is fed, which has to be the right materials.
Absolutely. This is, by the way, the thumbnail for those who would like to go to the why specific 20 amino acids in the Seeing Garden Biology series. And actually, I think in part of it, he has actually even gone to the alpha and beta amino acids and why it's biased towards the alpha, etc. It's really an amazing topic. So completely agree.
Yeah, let's move on here. Otherwise, so here we have the pathways. One is the de novo synthesis to make purine and pyrimidine bases, and then it goes down here, and here the salvage pathway, where purines and pyrimidines, they are by catabolic pathways, they are degraded and then recycled.
Amazing.
So here we have the sugar. This is the ribose 5 phosphate, in short ribose, and it is a product of the pentose phosphate pathway in all organisms. Why is it the pentose phosphate pathway? Because the sugar is a pentose, it has 5 carbons, and it has a phosphate moiety attached to it. And the pentose phosphate pathway transforms the glucose at the entrance of the pathway into the ribose 5 phosphate. Okay, so and where does it start? It starts with the reversed. That is, we have 6 different carbon fixation cycles. This is the reverse 3-carboxylic acid cycle, and it is supposed to be the primordial one, or the first one, which was starting and then diverged into the other 6 different cycles. The problem here is that, for example, in photosynthesis, which has another cycle, there are completely different enzymes like Rubisco employed. So if you claim that there was an evolutionary transition from the primordial TCA cycle to the cycle used in the Calvin cycle used in photosynthesis, how do you go from one completely different enzyme or set of enzymes to a different set of enzymes? That's like, for example, saying that we have a motorcycle made by one brand, and we have a completely different brand, and we need to evolve one motorcycle to a motorcycle from a completely different brand with different ingredients and parts. You can't do that. So this is a big problem for evolution. So the cycle starts here, and there are 9 different enzymes required to fix carbon dioxide to get acetyl CoA. That is the starting point to go to glucose and then to the ribose, which is used in nucleotides. So, of course, at the primordial Earth, we didn't have any enzymes whatsoever. So that's what I said previously. What they are supposing that there was a non-enzymatic carbon dioxide fixation cycle, which was on clay and minerals. And where do you get the energy from? We use here ATP as energy, which actually this cycle makes ATP. So we need ATP to make the enzymes which make that cycle. So you have a chicken and egg problem here. What came first?
Yeah.
The enzymes which make DNA or DNA which makes the enzymes? You need both. So you have a big problem here. And as well ATP, which is the product. So it is the same if you claim that there was energy somehow, like electric discharge events or hydrothermal vents or whatever kind of energy you want to suppose was active and in action there. How do you funnel it to have the precise reactions? It is like you have a gasoline, you put it and then you put it on fire. You have an explosion. You have an uncontrolled event of energy release. So this is a big problem.
Hello? Angelo?
Yeah. So what happens here is you go from the acetyl CoA derived from the reverse TCA cycle and convert it into pyruvate where you get 3 carbons. We see that here in the image. You go there down on the right side to the yellow acetyl CoA. You go to and feed acetyl CoA, transform it into pyruvate and feed the gluconeogenesis pathway. And through that pathway, you have here 10 enzymes, you get glucose. And that is the pathway that we know. So if you don't have this pathway prebiotically, how do you get all these enzymes? It's the same problem as in the reverse TCA cycle. If you don't have the enzymes, you can't have the reaction. You cannot transform pyruvate into glucose. And that is a big abiogenesis problem.
Hello?
You were kicked out.
Yeah, so something failed at my end, but did you go on or did you stop for a little bit?
No, I waited you coming back.
Okay. So let's go on. Sorry for that. Yeah.
Yeah, so I was explaining. So we go just to recapitulate.
Yeah.
Here we have the product acetyl CoA, and from that we make pyruvate. We go from a 2-carbon molecule, we transform it in a 3-carbon molecule, and it enters the gluconeogenesis, which is the reverse process from glycolysis, and we transform it into glucose using 10 enzymes in the pathway. Okay. Usually glucose is used to make energy, but in the reverse situation, we are making anabolic products, which is glucose, which then will enter into the let me check here. into the pentose phosphate pathway. So glycolysis is fed into the glucose is fed into the pentose phosphate pathway, and then it will transform it into ribose 5-phosphate, which is the sugar molecule of our purines of nucleotides, RNA and DNA. So you see here, we have here many different enzymes in that entire pathway which were not extant prebiotically. And we are making through this process just the ribose backbone of DNA and RNA.
Yeah.
Okay. So this is a huge abiogenesis problem. If you
Yeah.
don't have all that these metabolic pathways, which are like production line robots manufacturing things, how do you get ribose? It's a big problem.
Yeah. Yeah. And the one that you showed in the beginning is Krebs cycle, I think. And this is really a big issue because it's very fundamental and you need all those enzymes. So it's a big chicken and egg, as you say.
Big problem. And the other thing is some of these enzymes, Ahmed, they use like molybdenum sulfur and iron sulfur clusters, which are
Yeah.
inserted in the pocket to have the reactions. And when you go and study the complexity to make these metal clusters, it's just mind blowing.
Yeah.
Because you need to uptake these metals and sulfur from the environment, and you need to chelate iron to have it in liquid form to uptake it into the cell. So we
Wow.
are not stopping here. We need to have other machines that make these machines which are used in that process here.
Yeah.
So
The
it's
very basic and fundamental cycle, chemical cycle in life needs so many machines around it. It's completely impossible that it has originated from nowhere.
yeah, let's say that you have the machinery to import iron and sulfur. So
Yeah.
what do you make with that? So you have the materials inside of the cell. You need still a very complex biosynthesis pathway, also requiring several enzymes and proteins to make just a subunit of these enzymes used in that cycle. So
Yeah.
it is complexity over complexity. And the worst of all, Ahmed, is you don't have evolution at this stage because we are talking here about abiogenesis. We are
Yeah.
just talking here about making DNA and RNA, which is fundamental to have RNA and DNA replication and evolution. So
Yeah.
um, there is no hope for atheism.
Yeah. Yeah. Absolutely.
So let's move on. So
Mm.
here we have the nucleobases. Let's just show what we are talking about here. These parts here, the bases.
Mhm.
So we are going to show how these are made now. Okay.
Yeah.
So here, this is just the metabolism to have the pyrimidine nucleobases, which is a 6-member structure ring. This is the easier one, and you see how complex it is to make just the basis of
Yeah.
the
So
nucleotides.
those are just the bases which are fundamental for the genetic material itself.
Yes, to make the basis of the nucleotides.
Yeah.
It is very complex. Here is another graphic just to show how many ingredients and enzymes and machines that are employed to make these parts, to make these bases. It's mind-blowingly, staggeringly complex.
Yeah. Those bases are just the letters that are going to be used later just to write the DNA.
Yes, yes. We are talking just about cytosine, thymine, and uracil, which make up the letters of the nucleotides. So very small part actually, but very complex to make.
Yeah.
And here is the same problem. You have a huge discrepancy between the prebiotic models which try to explain how these parts could have emerged prebiotically by discharge and hydrothermal vents and meteorites and all these proposals. How do you go from that? Let's suppose you have these materials and they make the first RNA and DNA strand. Okay. So how do you go from there to make all this complex machinery, which then starts to actually synthesize and in a factory-like process, synthesizing and metabolizing these parts? It's like I forgot the name. She said that it is a bigger trajectory to go from an amino acid to a fully functional cell rather than to go from a fully functional cell to man. It's a bigger trajectory.
Absolutely. You know? So the point here you're saying is even those who are studying abiogenesis would try to figure out a way that those nucleobases could have existed in a prebiotic setup. But if you compare this setup to the setup that actually happens in the cell to make this a sustainable process, it's completely it's unmatched.
It's unrelated. Yes, it was Lynn Margulis. I remember now. She made that comparison, which I think is spot on. Because even if you have the explanation how it could have happened prebiotically, and you have the full set, even of the amino acids, the nucleotides, everything, okay, how do you go from that state of affairs to a full developed chemical factory which makes these building blocks through this complex machinery? It's a huge gap. It's a huge gap. And that's what I wanted to show here in that stream, so that people can have actually an idea, because that is rarely mentioned in abiogenesis conversations, what we are talking about and what kind of complexity that we are talking about. And
so
here
for the audience, let's just assume that we found in nature a piece of wood with a little graphite inside of it, so it can be used as a pencil. Okay, and then somebody tries to figure out a way how this is formed, and then when you compare it to a factory that produces pencils, you find that it's a completely different thing. So even if you prove that it is possible to have a pencil in nature by coincidence, you cannot extrapolate that on how a factory that produces real pencils that we use every day could have evolved, because obviously there is this one that you would like to try to find a path to make it happen by chance will not produce the factory that will produce 10,000 pencils every day. This is one animal, and this is a completely different animal.
Exactly.
Example.
You brought up a good analogy here. That's precisely what we are trying to explain here. Yeah.
All right.
So this is also here about purine, which is even more complex. It is a little bit. It is a two-ring structure of purine bases of adenine and guanine.
Yeah. So the bases that form the genetic material are either purines or pyrimidines. The pyrimidines have 3 letters in them, and the purines have those 2 letters. So we have the A and G here, and the CT and U in the other one. Yeah.
Right. Yeah. Here is just another schematic of the metabolic pathway to make the bases of purines. So it's very complex. Wow. Very complex. Yeah. So this here is purine synthesis starts with ribose 5-phosphate, which undergoes activation by ribose phosphate dephosphokinase to form PRPP. The product phosphoribosyl pyrophosphate acts as an essential component of the de novo synthesis of purines. This function of the enzyme would thereby undermine purine metabolism. So If you have a fully fledged, fully operational cell, and you don't have just this enzyme, nothing goes anymore.
It will collapse because no more
Yeah.
de novo nucleotides will be generated anyway. So
Yeah.
it will start and then it will stop and die, and that's it.
Exactly. What does that mean? That the cell is irreducibly complex. And if you have just this protein by itself, what is it good for?
It needs to be part of the complete factory with the feedstock and the output and the recycling. So it will do nothing again.
Yeah, exactly. That's the great point of irreducible complexity, that the single part by itself makes nothing. So why would natural mechanisms come up with such a complex structure which only has function inserted in the greater structure to have something more complex made?
Yeah. There is no drive behind it. Yeah.
Exactly. That's where naturalism simply breaks down. And it is very clear, very logical. Anyone can understand it. And you just have blinkers and don't understand it if you don't want to have God with its foot into the doors.
Yeah, absolutely.
So this is the Purine structure. And you see that you have several
Oops. Sorry. Yeah.
You have here several atoms in the ring structure, nitrogen, carbon, and so forth. And there are several different sources which are recruited to bring the atoms to the building site where it is constructed. So, like, for example, you need to have aspartate, which is an amino acid. You need to make the aspartate, the amino acids, that so it can feed the nitrogen atom in order to make the structure.
Yeah.
So here we have the biosynthesis pathway to make de novo purine bases. And so you have in total here 15 enzymes. Also, they have to be lined up in the right sequence. This is another problem. How do you get these enzymes in the cytosol lined up and ordered in the right sequence in order to start producing bases where one subproduct is handed over to the right next enzyme, which then continues the manufacturing process. And this is also something which science is studying it. How do they line up in an ordered way? This has to be precise and correct. It is the same as you have in a factory, an assembly line, and you need to put the robots in the right sequence in order that you can in the end have a functional subproduct. So this is a big challenge for abiogenesis to explain how that can emerge by unguided means. So here we have precisely that situation. Here they start with the single parts, the single enzymes here. Then they are joining into the core in the cytosol, in the liquid, and then they start to join together into a network where these reactions can take place in the right sequence.
Yeah.
How do you get that? How do you get that by unguided means? Trial and error. You try a million times until you have the right sequence. How do you get that?
Even with trial and error, it doesn't make sense, because just the word error means that you know what you want. For example, somebody was having a discussion with me about genetic algorithms that are used in computer simulations, and they said, humans use genetic algorithms that function based on trial and error. And the truth behind that is actually that we as humans define to the algorithm what is an error when the newly generated iteration diverges away from the required target. So trial and error only makes sense when there is an intelligent agent who defines what is the target, and then puts this, we call it in engineering, we call it a cost function. And then with each iteration, you recalculate the cost function, and you find whether you are getting closer or farther away from your target. So trial and error. If somebody says it's by trial and error, then he has immediately implied that there's an intelligent agent who is monitoring the process and directing it. Other than if you remove an intelligent agent from the formula, if there is no God in this case, the intelligent agent can only be God. Then you cannot even appeal to trial and error. You have to appeal to sheer chance. Just by chance, voila, it's there. It's completely ridiculous. I mean,
Yeah, I think you remember the experiment of Dawkins, where
yeah.
he said through evolutionary algorithms and programs coming to the end, sentence me thinks it is like a weasel. I don't know if you remember that. That was his target sentence.
Yeah.
And then he showed the program how in the end, by several cycles, you come to that sentence. But the thing is that things like a weasel is the target sentence. So,
Yes.
of course, you did set that from the beginning as the target where you want to go. But evolution, of course, doesn't have any goals, any target. So it
Yeah.
can go anywhere.
Absolutely.
Right?
So actually, even his example, unfortunately, when those people like Dawkins, who people would look to as a scientist, but actually what he is doing here is actually he's a science explainer actually. And the example he's using is completely defeating the purpose, because you are using a genetic algorithm that is programmed by an engineer that has a specific target, and it is
Exactly.
it is rowing towards the target. And then you are
Yes.
using this example to prove that there is no intelligence in creation. Nonsense.
Yeah, you are cheating and you are just fooling the uneducated audience, which just
Absolutely.
swallows whatever you are saying, right?
Absolutely. Unfortunately, it's an abuse to the trust of an audience when you give them an example that they cannot validate given their layman knowledge.
Exactly. Yeah.
Because it defeats the purpose from science explanation. If you do not explain what is a genetic algorithm and how it works, and then you give it as an example for the absence of intelligence, it is simply not an honest example, to say the least. Yeah.
I think these are very good examples to show through analogies what we are talking about. So anyone can actually understand it. It is very difficult to make a machine, but to make a lined up production line with several machines which work in a joint venture together, that of course is a much more complicated feat. And of course you need intelligence from the beginning to know what you actually try to achieve. What should the product of that production line be? And when you have that from the beginning, knowing what you want to achieve, then you can sit to the drawing table and make actually the design of each single machine. And then you need the full knowledge how to join them to have that end product in the end. If you don't have intelligence involved, you have the essence of what is necessary just removed, and blind chance simply will never get you to such complex things.
Absolutely. No doubt. Yeah. And this example that you're showing on the screen is essentially completely unbelievable that this Purinosome will will just happen. It's totally unbelievable.
Yeah. So here again is the entire biosynthesis pathway to have RNA. Okay. So here that is the pyrimidine ring, a single ring at the beginning, and then since it is just a one ring structure, it needs just 7 enzymes to make it, but the same, it is very complex. Okay. So since we don't have much time, I'll jump here to have a closer look just to one of these enzymes, which is carbamoyl phosphate, phosphatase. Okay. And it is very fascinating. There is a nice description just of that enzyme, which I will quick read here, just for the audience to have an idea about how complex just one of these enzymes actually is. Okay. So Dozens of enzymes are needed to make the DNA bases, cytosine and thymine, from their component atoms. The second step is performed by aspartate carbamoyl transferase. In bacteria, this enzyme controls the entire pathway. In human cells, the regulation is more complex, involving the interaction of several of the enzymes in the pathway. The enzyme, quick stop here. This is also very interesting, that the entire pathway is controlled by feedback loops. That means that the cell knows to control the production rate that it needs. And if you need, if you need, for example, a large quantity while you make the replication of a cell, you need a higher production of DNAs. So it gives the instruction to the machinery, make more nucleic acids. And when it is overproducing, it gives the signal, hey, slow down, it's too much, it's too many of them. So there is a factory regulation system in place here, which has, of course, also to be instantiated. And of course, that was not extant prebiotically. So you have a very fast degradation rate prebiotically of the naturally occurring nucleobases. So you would need constantly to overturn that and make more nucleobases to overturn the rate of nucleobases which are being degraded until you have a sufficient number that then you can concentrate and start actually the shuffling to then somehow having an RNA or DNA polymer structure. And how do you go from no regulation to the regulation of that system?
Again, the whole point of having an integrated system that self-corrects and that produces the right amounts of things and fits in the whole process, apart from the very complexity of the system itself, which is on its own totally amazing.
So let me keep reading here. The position of the two subunits relative one to another is critical. Take just a moment to ponder the immensity of this enzyme. The entire complex is composed of over 40,000 atoms, each of which plays a vital role. The handful of atoms that actually perform the chemical reaction are the central players, but they are not the only important atoms with the enzyme. Every atom plays a supporting pan. The atoms lining the surfaces between subunits are chosen to complement one another exactly to orchestrate the shifting regulatory motions. The atoms covering the surface are carefully picked to interact optimally with water, ensuring that the enzyme doesn't form a pasty aggregate, but remains an individual floating factory. And the thousands of interior atoms are chosen to fit like a jigsaw puzzle, interlocking into the sturdy framework. Aspartate carbamoyl transferase is fully as complex as any fine automobile in our familiar world.
Wow. The very idea that it has 40,000 atoms is just amazing. 40,000.
Just one. And if you take the smallest free living cell that we know today, it is a bacteria called Pelagibacter rubic, and it has 1,350 proteins. So you have 1,350 machines similar in complexity to this one, which must be just right. And you need to interact them together. And James Tour has brought up the interactome, which is precisely what we are talking about. It's not enough that you just have the proteins. You need to have them interacting together in the right way. They work in a joint venture together. So you need to interconnect them in the right way. How do you get that without intelligence?
It's totally impossible. Great example. And
And in the pathway, I show you here another one that is OMP decarboxylase, and it is known for being an extraordinarily efficient catalyst capable of accelerating the uncatalyzed reaction rate by an impressive factor of 10 to the 17th power.
Wow.
That means that The non-catalyzed reaction takes 78 million years.
It's totally outrageous. Yeah.
This is the second fastest enzyme that is known.
Wow. Yeah. And if I actually have I have this evolution course series, and in one of them I address this matter of the enzymes. And it's just completely amazing. So if somebody says, well, enzymes are just catalysts, and maybe the initial reactions in the prebiotic world happen without the enzymes. And then when you look at those figures, you know that's completely ridiculous, because one reaction will take few million years, and the other components that will complete the chain will not just be sitting there waiting. We will wait for it to finish. They will go away, or they will degrade, or even the very ingredients of the reaction itself that needed the catalyst will go away or degrade or combine with something else naturally.
Depending on the temperature, we know that the nucleobases or the ribose, they have a degradation time of days to a few years.
Yeah.
So did they wait 78 million years?
We are patient. We are patient chemicals. And they die and others come in their place, or actually they decompose because they are not living anyway. So
right.
Yeah,
right.
It's, totally. When you see the numbers, it makes you feel really the how absurd the arguments for this happening on its own is really the case. Yeah.
Yes.
Yeah.
Now, I think we are running out of time, but let me show you this one. This is a picture of ribonucleotide reductase.
Mm-hmm.
This is a monstrous enzyme, and it transforms RNA into DNA. Okay. There are three different configurations: class 1, class 2, and class 3. They make the same reaction, but in different ways.
Mm-hmm.
And it is a very complex enzyme. And if you don't have that enzyme, you don't have DNA.
Wow. Yeah. It converts what into DNA?
Let me go to the beginning here.
Yeah.
Here. This small part here, in RNA, we have a carboxyl moiety, which is removed. The hydrogen is removed. Actually, sorry, the oxygen is removed from the moiety here, and you have just hydrogen left. So it is just removing an oxygen atom.
Ah, okay. So it becomes deoxyribose instead of
exactly. It deoxidizes ribose. It removes oxygen from the position here. So that seems to be a very small thing, but it is essential in order to have longevity of DNA. And you need a monstrously complex enzyme to achieve this. And if you don't have RNR enzymes, you have no life.
Yeah.
So I cannot go in a short live stream here into the details, but here we have, for example, other enzymes which charge the enzyme in order to make the reaction. The energy is constant. It is constantly recharged in order to perform its reaction. So you need at least the two to have the function.
Yeah.
Without recharging, it cannot work. So how do you get that?
Yeah. Amazing. And if you look even at RNA, it's a very unstable thing. So life could not have started with RNA because it mutates like crazy, like 1,000,000 times more than DNA. And because it's unstable and there aren't so many error correction mechanisms on it, or actually very few, because it degrades anyway. So if if life did not have a pre-engineered mechanism, even if somebody says, well, it started with RNA and then went to DNA, it means that you assume that the mechanism to make this conversion in a very efficient way also existed through an intelligent agent, because there is no other way. So, yeah.
I mean, if you just study RNR enzymes, and I am sure there are scientists which are studying just that enzyme for their entire life.
Yeah.
For so complex it is,
Yeah.
And
Yeah.
here you have just one of the metal clusters which is used, which also has to be synthesized by very complex manufacturing pathways.
Yeah.
So you
Yeah.
need machines to make machines, which make the building blocks of machines.
Yeah. Yeah. And then the question, where did all those machines come from? If those machines are needed to convert RNA to DNA, for example, and those machines need to be coded somewhere in another genetic material, they cannot be coded in RNA, because RNA will not survive for so long enough. And then it means that DNA had to pre-exist to create the machines that will convert RNA to DNA, which is a
Exactly.
which is again,
It's again a chicken and egg situation.
yeah,
We
the
need we
whole,
need
yeah,
we need RNR enzymes to make DNA, but
yeah,
we need DNA to make RNR enzymes. Which came
absolutely,
first?
yeah, absolutely, yeah. It's if you remove the intelligent agent, which is essentially God, from the formula, then you only end up with paradoxes coming from chicken and egg situations. Yeah, absolutely.
Yeah.
Yeah.
This here is one of the cofactors of the three kinds I used, which is a vitamin B12.
Hmm.
And vitamin B12 is the most complex vitamin to be synthesized.
Hmm.
So in order to have one of the three classes of RNR enzymes, you need to have that cofactor, and it is about 30 different enzymatic steps to make just that cofactor.
Yeah. Wow. How many again?
I think 29.
29. Wow. Amazing.
Yeah. Yeah, so with that ends my presentation.
That was a wonderful and exciting presentation, Otangelo. So thank you for that. There is plenty of further reading for those who like to pursue this. I'm sure those with more curiosity can go and further investigate it. But the message is, life is so complex. It is almost unimaginable that it begins without very, 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 very careful planning and design. And obviously, it's even more unimaginable that if it starts and then continues, that it is by chance, because you need all those integrated cycles that will keep the machine going and then create integrated kinds of life that will help each other and live in symbiosis. And dead material becomes food for living things, etc. And living things are food for living things, and living things complement living things and fix the environment and fix chemicals. So it's a wonderful world. So thank God we believe in God, and I hope that people who are atheists and who do not believe have a good stand with themselves and look in the mirror and find out a way how this person in the mirror could have been a rock one day without a creator. doesn't seem reasonable to me at all. So thank you for you, Otangelo, for this presentation and for coming here. And for everyone who's watching this, by the way, I will be resuming the series with Dr. Fazrana soon enough. We'll be recording more of the more material and putting it, let's say, part 2 of Seeing God in Biology will be coming soon. We'll be announcing that, God willing, inshallah, pretty soon. And thank you, Otangelo, very much for being here and sharing your nice presentation. I wish you plenty of luck for your book, inshallah.
Yeah, it will take still some time, but I'm working on it, and I hope that soon I can publish it.
Yeah, maybe we can do a book review together one day.
Sure. Okay.
Yeah. So thank you, everybody. I'll be putting the extra now. Please share this with those who are interested in the topic, and see you again soon. Probably I'll also be restarting my Jewels series on Thursdays. I'll also be announcing that soon. And God bless you all. Thank you, Otangelo. Thank you for everybody who's watched this. And please don't forget to like and share the video. And I'll also be putting a couple of links for Otangelo's website and YouTube channel in the description for those who would like to have a look there for more of his work. All right. Thank you all.
Bye-bye.
Bye-bye.